HIV is incurable due to its persistence in cellular and anatomical reservoirs. This method allows an HIV reservoir assessment of immune cells isolated from the lungs.
This technique allows the isolation of pulmonary mucosal T cells and alveolar macrophages from bowel fluid for further phenotypic or virologic assessment as cellular reservoirs of HIV. Understanding the biology of alveolar macrophages and CD4 positive T cells and their contribution to the HIV reservoir could lead to important insights into the challenges facing an HIV cure.
The isolation of primary macrophages from bowel can be applied to various other research areas, including inflammatory or infectious pulmonary diseases such as asthma and tuberculosis.
After obtaining the bowel fluid from a human patient according to standard protocols, vortex the fluid in the original collection tube before transferring the sample to a 50 milliliter tube within a biosafety cabinet. If the fluid appears very turbid or contaminated by filamentous tissue, filter the sample through a 70 micrometer nylon mesh filter into a new 50 milliliter tube. After centrifugation, transfer the supernatant to a new 50 milliliter tube and use a pipette tip to gently break up the pellet containing the whole bowel cells. Resuspend the pellet in 1 milliliter of RPMI 1640 medium and add 1 milliliter of the reserved supernatant of each of 10 1.5 milliliter microcentrifuge tubes. Add 10 milliliter aliquots of